Okay, welcome to the lesson uh, devoted to proteins. With the first part, we are going to talk about the basics, so the structure of building blocks of proteins, so amino acids, then the peptides, so how the amino acids join together and what structures do they form, and then the differences in the two main types of proteins, fibers and globular. On the separate lesson, we are going to talk about enzymes, the structure and role, and then we will explain what happens in enzymatic reaction, describing the rate and factors affecting the speed of the enzyme. Okay, what do we know about the proteins? What do we have to know about proteins? The proteins are made of amino acids. This group of organic compounds, I mean proteins, is the most variable the group of substances because they building blocks. Amino acid they have different properties, and they may be arranged in different sequences, giving the unlimited variability of proteins. But let's first focus at the amino acids alone. Amino acid, as the name says, has the amine group. You can see it here on the left, and the group of organic acids, so carboxyl group, here it's seen on the right. Okay, the center of compound is a central carbon with hydrogen and the R group. The R group is a variable group, meaning that different types of compounds may be attached, giving the amino acids unique properties, such as some amino acids in this area may be non-polar, some of them may be polar, some of them will be acidic, the other ones will be alkaline. But in general, each amino acid is soluble in water because amine group, as you know, has a slightly positive charge here, okay, because it can even touch additional hydrogen, whereas this hydrogen may quite easily detach, so the negative charge may appear at this end of the amino acid. That's why very frequently amino acids are used in chemistry as buffer substances that will keep the constant pH for enzymatic, for chemical reaction. Okay, in the case of the R group, we distinguish 20 different groups, therefore 20 different amino acids important for living organisms. Of course, there are more amino acids in organic chemistry. For organic chemistry and biology, we need only 20 of them. Okay, how do they join? On the previous lessons about lipids or carbohydrates, you've learned about the condensation reaction. The condensation reaction happens as well in here. So the amino acid carboxyl group links with amine group of another amino acid, and they form a characteristic bond called the peptide bonds. As in all condensation reactions, water is a byproduct. And a compound made of, a molecule made of two amino acids is now called dipeptide. And they can, for the condensation may continue, and longer chains will be produced. For instance, here you can see the molecule made of three peptides, and we can identify the peptide bonds is here and here, okay, between carbon and nitrogen. Okay. In the books, you may find different names for peptides, proteins, and the question was the difference. If chain is made of 2 to 20 amino acids, we call it oligopeptide. The polypeptide, it's a word reserved to a chain of 20 to 100 amino acids, whereas protein is a word we use to describe longest chains above 100 amino acids. But what is more important in case of a protein, this name suggests that it is functionally active. Okay, so polypeptide is just sorry, I have to raise it. Polypeptide is basically a chain of amino acids, but protein is also a polypeptide, very frequently longer than 100 amino acids, but with a function. That's the most important distinction. Okay, so let's take a look how this huge variety of proteins are made. All right, the structure of a protein is described by the levels of structure. The first level, it's primary structure of a protein. It's a number of and sequence of amino acids in one chain. So if we take a look at two peptides, here you can see 1, 2, 3, 4, and 5 with names: serine, leucine, alanine, glycine, and cysteine. And the other one, in which these two are in different positions, it's the fact that they are in different places makes them different. So there are two different peptides because they will arrange in different way when it will be placed in, for instance, water solution. But what do they have in common are the peptide bonds between 1, 2, 3, and 4. Okay, position of each amino acid in a chain is determined by genes. In a gene, the 3 nucleotides or in DNA code for the position and type of amino acid found in polypeptide chain. Therefore, if we take insulin, which is a polypeptide protein made of 50 amino acids, it means that the number of nucleotides coding for this protein is 3 nucleotides for initiation of a protein synthesis, plus 150 nucleotides of this 50 coding for this 50 amino acids, and 3 nucleotides to stop insulin production. This, what is in here, will be extended when we will start talking about processes involving DNA. Okay. What is specific in variety of amino acid sequences in living things? Nearly all living organisms use the same amino acids, and moreover, they use the same triplets, the same codons in genetic material to code for the proteins. However, there are some exceptions, and these exceptions will be shown later, because today we talk about protein structure. The second level of structure of a protein is secondary structure. This structure is formed when polypeptide chain, here you can see the polypeptide chain, coils and will be stabilized by hydrogen bonds. Hydrogen bonds between this hydrogen, for instance, and oxygen in here. So nitrogen of amine group and oxygen from carboxyl group. But what is specific, it is within the chain, with one polypeptide chain. This coiled chain, because it has a helical shape, is called the alpha helix. The other regular structure that may form in a polypeptide chain is a beta-pleated sheet. The beta-pleated sheet is also held by hydrogen bonds. However, here we can see that it is more flat, not coiled. All right. Okay. In case of next level, so tertiary structure of a protein, this is the spatial orientation of alpha helix, okay, in a parallel way or in a spherical way. And therefore, we distinguish the fibrous proteins and globular proteins. Fibrous proteins are important for living organisms as structural components, such as collagen. Collagen is a most abundant protein in animals, and it is found in our bones, in our nails, in our skin, because it is very resistant to stretch. Globular proteins, here you can see the example, the myoglobin, which is a protein in muscles responsible for storing oxygen, is coiled in a spherical way. So that's why it forms, we call them a globular, so globin. And its solubility in water will be relatively high, higher than the collagen. Globular proteins, because of their solubility, will have a different range of roles. What links are responsible for maintaining the tertiary structure of a protein? Here is a quite high variety of links. The one type of bonds, the ones that we already know from the molecular biology, are hydrogen bonds. Hydrogen bonds, but this time they are made between the R groups. Here you can see a one R group and the other R group. Okay, so not between amine groups and carboxyl groups, but between R groups of an amino acid. The Second type of bond is a hydrophobic interaction, formerly known as the van der Waals interactions, but today we just describe them as hydrophobic interactions. We have to remember that most proteins in living organisms are dissolved in water environments. Water environment, as we know, is polar environment. So any substance that is polar can dissolve. So if a side chain is hydrophobic, so it repels water, it will likely attract another non-polar side chain. The third type of bonds between the polypeptide fragments is a disulfide bridge. It is a bond between the sulfur and sulfur of two different side chains. So the two different R groups. We find it in, for instance, cysteine. You probably experienced the existence of disulfide bridges, because when protein breaks down, for instance, when the white of an egg decays, we can feel the characteristic smell, the smell of hydrogen sulfides, a gas which is released because the disulfide bridges are broken. The last one, the fourth type of bonds in tertiary structure, is ionic bonds. Again, the side chains, some of them may have a strong positive charge, and the other part, the other chain, may have a negative charge. And because of the electronegativity, well, these details are used in chemistry, the ionic bond may form. So this hydrogen from hydroxyl group jumped to link to attach to this nitrogen. Some proteins, not all of them, but some proteins have also the quaternary structure. The quaternary structure is reserved to complex proteins, proteins made of not one but many chains. Moreover, very frequently with non-protein component. A very good example of a protein of this with this structure is a hemoglobin, which is made of 4 globins, so 4 spherical polypeptides, and there are 2 alpha chains and 2 beta chains. Each chain has separately a heme group that traps oxygen with iron, and we find it in our blood. Okay. However, these bonds are susceptible to some certain environmental factors. For instance, when high temperature, unfavorable pH, too acidic or too alkaline, or strong alcohols act on a protein, the protein denaturates. And denaturation of a protein is connected with breakdown of quaternary structure. Of course, if a protein has that level of structure, but tertiary and secondary structure may be broken as well. In most cases, primary structure is kept unless the protein is burnt. But when, for instance, when the egg is boiled, all the levels of protein structures except from primary structure are destroyed. To some extent, protein denaturation may be irreversible, but it is only when this change in shape of a protein was just very slight. If the change is very, very large, this process is unfortunately irreversible. the last topic for today, functions of proteins. Because of the properties of globular and fibrous proteins, we distinguish different functions of the proteins. The fact that fibrous protein is water insoluble determines the functions it can play and functions it cannot play. For instance, the structural proteins such as collagen, keratin, or spider silk are made of fibers of alpha helices, chains linked parallelly and able to withstand the stretch. However, they will not dissolve in, for instance, plasma. Whereas the globular proteins, because they are water soluble, okay, and they are folded in a spherical shape, they can be transported inside organisms, dissolve in cytoplasm, in this way having a variety of functions. For instance, they may be enzymatic, such as amylase in our digestive system or rubisco in plants. They may be regulatory, such as hormones, our famous insulin. They may perform some form of transport, for instance, hemoglobin in our blood. They may protect us, immunoglobulins found in blood, so the antibodies we use to fight diseases. Finally, they may be signal proteins, such as a rhodopsin in the retina of eye. We mentioned that protein and the retina when we were talking about stem cells. One more thing must be added to our lesson about proteins. It's proteomes and proteomics. What's a proteome? A proteome word very similar to genome. Probably you've heard about genome, which is the total DNA in an organism. It is a set of proteins of an individual. What is specific to proteome? Identical twins may have exactly the same genome. However, proteome is always unique to an individual. Even identical twins. So, for instance, when the forensic investigation is done and we want to check the fact whether one of the twins, who from the twins was for instance, a doer of some crime, we look for proteins as well. Yeah, proteins characteristic to an individual. Moreover, the proteome changes in our lifetime because some genes are expressed when we are child, some are expressed when we are adult. So the proteome is a dynamic set of proteins produced by an organism. So changes in gene expression. It makes us dynamically unique. The other thing, proteomics, is basically a study of proteomes. Biologists, molecular biologists who studied unique nature of proteins, because, for instance, even if the insulin is produced in individual A and individual B, some amino acids may be different in the polypeptide chain, but the insulin may still have the same function. And this is the area of study of proteomics. At the end, I have an exercise for you, and please submit this answer when you do your homework. The possible number of different polypeptides can be estimated, assembled is given by a P equals A to power of N, where A is the number of different types of amino acids available, and N is the number of amino acid residues in the polypeptide molecule. Okay, so if we know that we have 20 different amino acids, please calculate how many different proteins will be formed potentially in a polypeptide or peptide actually made of 5 amino acids, 25, and respectively 50 amino acids. Thank you for your attention.